lab
Okay.
on Thursday because I think we're going to have time.
Okay.
All right. Today we are talking about coral snake mimicry and two papers. Brody, 1993, Brody and Jensen, 1995. We will take Brody, 1993 first, and that will be Mason. Go, Mason.
Okay. So the Brody 1993 paper, the differential avoidance of coral snake banded patterns by free-ranging avian predators in Costa Rica. So I'll start it off by asking, I guess, the group why exactly Brody chose to use coral snakes in this study of, yeah, why coral snakes?
Because they have different banding patterns, and some of them are venomous and some aren't.
Okay. So
Are all coral snakes venomous?
I didn't think so. I thought there were some that. I'm looking at the figure 1, and it says non-venomous.
Yeah, those are not coral snakes, though.
Oh,
coral
my
snakes.
bad.
I think
those are the mimics, yeah.
Yeah.
Okay, gotcha.
Yep.
That's what I
So
meant.
for
Yeah, the
aposematic
mimics.
reasons.
There you
right?
go.
Yeah.
Yep.
Aposematic
Good.
reasons. So in the previous models mentioned in the introduction, mainly the first paragraph, why would they be questions for applicability to real models in nature? Like, what are the main issues with the previous studies? Or what makes them difficult to study?
They were caged.
Right. So.
It's not like as natural as like if they were in nature.
Right. So they were observed in unnatural ways according to how they'd be seen in nature. So why couldn't they necessarily just go and study predator-prey interactions in the field? What are the reasons? Difficulty of reasoning behind that. They don't really mention it. That question was probably worded in a way that was hard to answer, but what I was getting at is that prey and predator interactions in the field are really hard to observe because it's kind of based on luck.
I mean, they do mention it. It's the first sentence of the entire paper.
Right, but they... yeah, okay.
So, there is that.
Right, but you'd have to spend an ungodly amount of hours in the field, and it's kind of based on luck, seeing, a bird eat a snake, as we rarely see it. So then my fourth question would be, later in the introduction, they go on, Brody talks about these two predictions that remain untested. What exactly were these two predictions that he touches on?
Like, should dangerous models protect like a wide range of mimics, and are there like certain advantages to different patterns? Like, are some more effective than others?
Okay. So if we could flip to the first figure, Elizabeth already kind of examined it. It's examining the various color patterns and whether snakes are venomous or not and their species. According to these patterns and their and how venomous they are, can you guys point out whether you see Batesian or Malarian mimicry? Like for a specific color pattern, would you be able to point me out that?
Aren't coral snakes like the Mimicry and ones like that?
Are they what?
Like, that's their
Yeah.
scientific name.
Micrurus. Anything in the
Micrurus.
genus Micrurus is a coral snake. Yep.
So like any not with that name that don't have venom would be the Batesian, because they're a model and mimic that aren't dangerous. And then Malarian is where they're both dangerous, so like black and red and black and white.
Right. So,
So why don't we just to be really clear, take them from top to bottom.
Okay. So like
Brown gray, what do we have in there?
So brown gray, there's like you mean according to Batesian Malarian or like
Batesian Malarian, and
Venomous?
also pay attention kind of to level of venom. So this is something I don't expect you guys to know. There are a variety of different kinds of venomous snakes. There's a group of snakes that includes the king snakes, for example, now I've lost the family name. The colubrity. The colubrity, for a long time, was this big catch-all family of snakes. When you didn't know where to put a snake taxonomically, you just shoved it in the colubrity. And years ago, some people went through and kind of teased that apart and revealed that to be just a mess, and have been working on cleaning that up. But within the colubrity, which generally includes non-venomous snakes, there are certain species that have developed what we call a rear-fanged condition. They have a fang that is way back in the mouth, and they are mildly venomous. And the ringneck snake here in Missouri is one of these. It's one of the rear-fanged colubrids. But it has such a small mouth, and the fang is way in the back of the mouth, and the venom is so weak that we don't normally think of them as venomous. Although they might have venom that acts better on invertebrates than it does on vertebrates, et cetera, et cetera. Those are the species that are characterized with a single plus in the figure. So the pluses are rear-fanged colubrids, and that's what they mean. It's species that are related to the king snakes and whatnot, but their fangs are not particularly effective, and their venom is not particularly bad. The Elapids are the family of snakes that contain all of the coral snakes. It also contains cobras, and depending on whose taxonomic breakdown you're looking at, it also includes all the sea snakes, which are very venomous. So Elapids have front fangs, and their venom is what we refer to as a neurotoxic venom. Tends to paralyze the prey, as opposed to a hemolytic venom, which pit vipers have, which breaks down tissues. And then the non-venomous colubrids are things like the king snake that don't have any specialized tooth modifications and don't have a venom gland at all. So there are, in this figure, there are levels of venomous, from not at all to a little bit to a lot, and that's what they're trying to incorporate in here. So if we start at the top with the brown gray snakes, what do we have?
Question.
Yeah.
So like the black and white version where it's like a double plus and then a regular plus.
Yeah.
Would that be considered Batesian or Malarian? Like, are the plus really dangerous, or are they not?
Yeah, I mean, they're considering those to be venomous. They're just not as venomous as an actual coral snake. Mm-hmm. So I would classify that definitely as Mullerian. All right, start at brown gray, work your way down. Anybody can do this.
Batesian, you have your dangerous model and then non-dangerous mimics as well.
But you also have within that.
Yeah. You also have a dangerous radiate mimic.
Radinia and Coniophanes.
I'm assuming that the Coniophanes or whatever, and the Radinia are Malarian.
Yeah, so you have a mixture of Mullerian and Batesian in that color pattern. Mm-hmm. But remember, the brown and gray color pattern, the two Mullerian mimics are on the not very venomous side of things. What about the colored pattern?
Actually, quick question. Would that be considered mimicry if they're just a brown gray?
Yeah, those are like the.
It seems like a terrible color pattern to mimic, but yeah, I mean, in theory, if brown gray confers any advantage, which of course we see later,
But it doesn't.
It doesn't really. If it did, then that would be considered mimetic. But now you're jumping ahead.
Sorry.
No, that's fine. Because obviously, if you're going to mimic something, you would be doing it because there would be some mimetic advantage.
Yeah.
Mm-hmm.
So, all right, the colored pattern.
Then you have Mullerian and Batesian in that one as well.
In the color?
It would just be Batesian, right?
Just Batesian because you have a non-venomous species mimicking presumably a venomous species. In the collared pattern, second
Oh,
one
yeah.
down.
Yep, my bad.
No, that's fine.
I was looking at the wrong one.
Reverse tricolor.
No mimicry?
Well, no, because that's a pattern that is presumably mimicking what?
Oh, a coral snake, I
The
guess.
tricolor.
The tricolor. So it's a reverse tricolor. It's just slightly off. Instead of red on yellow, it's red on black. So
it'd
it's
be
a
Batesian
similar.
again.
So that would be a Batesian mimicry of another color pattern, which would be the tricolor. Then within the tricolor, you have.
Malarian.
And Batesian.
Yep, you have both Mullerian and Batesian.
Mhm.
Batesian both because of Scaphia odontophus and because of Lamprofeltus triangulum.
And then black-red is all Malarian.
Black and red is all Mullerian.
All venomous, and same with black and white.
Same with black and white.
And black-white with red stripe.
Black-white with red stripe might be actually Mullerian mimicry of the black and white, and vice versa, black and white pattern. Yeah, so it's useful to think of those patterns as whether or not Batesian or Mullerian mimicry are going on in any of those conditions. Continue.
Yeah, and then within this Micurus, I say that right?
Micrurus?
Micrurus species, they mentioned the word sympatric multiple times. Just to clarify, what does sympatric species mean?
Species that were brought up in like the same location.
Right.
Allopatric would be different
Mhm.
locations or or geographically restricted, I guess.
Right.
Not
s-
geographically
or,
restricted.
Sorry, sexually restricted as well.
Hmm?
Like reproductively.
Nope.
restricted. No.
Like they're
Sympatric refers to two species that have the same geographic range, so allopatric would be
Separate geographic ranges.
non-overlapping ranges.
Right.
Yep, yep.
So
You were
the-
you were correct on the first half, but confused on the allopatric part.
So, with respect to their big overall question of analyzing like free-ranging predators, why would it be important for the species to be sympatric rather than allopatric? For their overall test.
Species of snakes?
Yeah.
Like because you'd want them to all be like the birds to be familiar with all those
Mhm,
color
exactly.
patterns.
Yeah. And then on the right side of this page, it goes on to say towards the end what is he basically planning to do, and just to reiterate, what is the importance, or why is he choosing to use plasticine replicas rather than anything else to analyze predation of the different snakes?
So he's going to use the plasticine models of various color patterns of the venomous and the mimic snakes. He's using the plasticine because it's non-toxic to the birds, and because you can like see the imprint of the birds
Yeah.
in it, like we're doing with the caterpillars.
Yep. Okay, so on the next page, in this figure number 2, what exactly is going on? Like, with respect to experiment number 1, what is, it's kind of a big overarching question.
They compared the brown and tricolored, and the purpose of the white paper and the natural background was to get rid of crypsis to see if the tricolor gave them any type of like camouflaging advantage
Nice.
there.
So they're basically ruling out crypsis over
Yes.
their avoidance behavior.
Yeah.
Yep. And sticking with that, we can move to figure number 4, which is basically the results of figure number 2. And that's basically what Avery mentioned. It's pretty obvious what's going on here, and that it doesn't matter what background it is, the crypsis is not taking place, and particularly the behavioral avoidance is the main driver of that.
So somebody speak to me exactly why that pattern shows that it's not crypsis in
Right.
figure 4.
Well, it shows that against the natural and against the white background, the number of attacks for the tricolor was less than the number of attacks for the brown. So you would think that if there was some sort of crypsis going on, the number of attacks from the natural versus the white would be higher, like on the white background, because they would be more easily seen than if they were on a natural background. But those numbers aren't changed, so you can rule that out.
Yes. Whenever you see something like that, it's oftentimes useful to say, okay, they're claiming this supports the hypothesis that this is an aposematic color pattern rather than a cryptic color pattern. What would the data have looked like if it was actually cryptic? And then that helps you ascertain for yourself, is what they're telling you really consistent with how you're understanding that? That's a very good way of thinking about it. Good.
Okay, and in regards to their experiment number 2, we can shift attention to figure 5. Basically, for number 2, they wanted to analyze their relative mimetic advantage among all the clay models. And can someone tell me what's going on in figure A? Like, what is this a depiction of?
You're looking at the number of non-avian disturbances to see if there's a difference between, like, the number of times something non-bird related happened to the snakes.
So what, you mentioned non-bird related, but more specifically,
Just
what
like,
does non-avian mean?
I think they mentioned, like, being stepped on by a person, like being moved, like branches falling on them, stuff like that. That was, like, not an attack from a bird.
So in regards to predation, sorry, I'm digging even further. I'm looking for something specific. In regards to predation, what is predation of the snakes, non-avian? I guess what I was getting at is that these disturbances are from predators that are unlikely to be predators of snakes. Right. So with respect to where they were disturbed on the end and middle, what do we see within that?
For non-avian, it's inconsistent.
Right, it's inconsistent. And then moving down to the number of avian attacks, what are the black little bars in that graph B?
Attacks on the ends, or one of the ends, I think is what they mentioned.
Sorry, I missed the little bars above at the
Sorry.
top. Yeah, no, I'd be more specific.
Like significance in terms of like them being the same. So the brown is its own significantly different group, and then the first 4 banded patterns are different than the last 4, although the middle 2 banded patterns are significant in both groups.
Right. So, based off of this graph, can you specifically say, in answering the overarching question, does it matter which mimetic pattern the snakes have in order to avoid predation?
Not really. I mean, there was a difference in the groups, but it was like a 1% different, I think.
Right.
And those differences are really broad. There's a lot of broad overlap in all of those patterns once you get away from brown, right?
So to answer their overall question of experiment number 2, what would you say about relative mimetic advantage in regards to the 6 different colors that they used?
There's not really one for the banded.
Right. Yeah. So the brown just is so much more statistically different that basically these birds are they're avoiding rather than making a little mind map of which color is Batesian or Malarian and which one they can attack due to their risk reward, basically. So, and then moving on, they mentioned.
Before we leave figure 5B, what else distinguishes 5B from 5A?
Targets on the ends of the snakes?
Yeah, targets the ends of the snakes primarily in 5B. So avian predators, when they attack a snake, were you about to go to this? If you were.
No, yeah.
My
you got it.
apologies.
No, it's okay.
So when the birds attack them, they're attacking mainly on the end of the snake, which in theory would be the head, as opposed to with the non-avian attacks, there was no pattern between head and middle of the body. So it appears that, you know, snakes are recognizing these things as potentially live things that are dangerous, and if you're going to avoid being struck by a snake, if you've ever picked up a snake, if you pick it up from the head end, you have control of the head. If you pick it up from the middle of the body, it comes around and bites you. And so they seem to be treating these things as if they are actually real snakes in terms of where they're attacking and the frequencies of attacks on the different color patterns. And that's one of the questions you really have to ask about a study like this is, these are clay models. Are the intended subjects of your study actually perceiving them as something other than a clay model? And so if you have data that speaks to that, it's good to note it.
And there was a mistake in Figure 5. I don't know if any of you guys caught that. It threw me for a loop. I actually had to go talk to Paul about it. But they basically they mentioned the solid bars indicate the number of attacks on the middle, and the stippled indicate the number of attacks on the distal portions, and is actually flipped in the legend. But
yeah, and the legend is correct, and the verbal description in the caption is not.
Yeah.
And the only reason you know that is when you go down and you look at the patterns of head versus middle of the body strikes in the statistical analysis, tells you that the wording in the figure is incorrect. And they never published an erratum correcting that, but oftentimes those things slip through in the publishing process and nobody ever picks up on it.
Honestly, the stippled thing was kind of hard to see anyway.
Yeah, it
It
was.
looks just like.
Yeah, you have to see the original printed version. I went and got the
Okay.
printed version for Mason so he could look at that.
Yeah, it did help. Yeah.
The top white, what appears as white, is actually stippled in the original.
Okay, so moving on. In their discussion, they mentioned their plasticine replicas. So my overarching question about the replicas is, what were the potential pitfalls mentioned in the replicas? Or what could cause the data to be altered due to the behavior of the models?
This may not be what you're asking, but they mentioned that they had to mark multiple similar looking disturbances as one, because they concluded that it was part of one single attack event. So given that they track everything, it could be a bit confusing and inaccurate if they were to try to strike all those out and narrow them down.
Yeah, so they're being conservative here. So rather than counting the number of marks on a model, they're basically saying, regardless of number of marks, was this model attacked or not? And that's a conservative way of going about it, because you can't determine whether all those marks were made by a single bird attacking the thing a bunch of times, or a bunch of different birds attacking it at different times. And we've seen this with models that we have put out in the field here. You'll see a model that is bit into, and it's bent, and it you can tell that a bird is really tied into this thing before it realized that it wasn't worth being tied into. And so that thing is really damaged. There's a lot of damage. It's destroyed, basically. There's no salvaging it to use it over again in an experiment. But chances are that's probably just one really hungry bird that latched onto that thing and did all of this damage. And so that's a reasonable kind of conclusion. And it's on the conservative side, because you don't start racking up lots of instances of attacks because you're counting each individual mark on a given model. So it's just a conservative way of going about doing it. But yeah, I don't know that was what Mason was headed for, though.
I think what you were asking about is if the birds were going to learn if the replicas were inedible, or if they're going to learn that they weren't prey over the course of the
Partly,
experiment.
yeah, that was part of it. The main part for the discussion of the model efficacy was in the last paragraph of page 232.
Yeah, that's where I thought you were headed.
Mm-hmm. So according to their
Well, because of like their lack of anti-predator
right,
behavior.
yeah. Why could that cause? So due to their lack of behavior, what would be the reason that Brody was getting at that could potentially change their data?
I mean, I think you'd be saying like the snake doesn't fight back, so that results in like them seeing a beak imprint from an attack, but like the snake isn't behaving how it usually would.
Mm-hmm.
And as a result, you might, with a real snake, you might have never seen that
Yeah.
attack, and so it might be actually an overestimate of the number of attacks
Mm-hmm.
if the snake models could behave.
Mm-hmm. But for the latter half of the paragraph, does he deem this to be an issue for this particular study? No, why not?
Because all the replicas had like the same potential to attract or repel
Exactly, yeah.
predators.
Yep, so even though they can't run away or, behaviorally fight off predators, they still have the opportunity to attract or repel them based off of their color, which is what he was wanting to get at.
Yeah, this is a common rhetorical device in a scientific paper. You bring up a potential criticism to the results, and then you explain why that criticism isn't valid. And that's exactly what they're doing here, saying, this could be an overestimate of the number of attacks, but what really matters isn't the total number of attacks. What matters is the number of attacks relative to other replicas, and all those replicas are the same in that none of them have behavior.
Mm-hmm.
And so, this actually gets to some fundamentals that you guys learned when you were first years. At the crux of it, what's the difference between a control group and an experimental group in an experiment? So this is a rigid kind of experiment. It's a manipulative experiment. What's being manipulated?
The color pattern.
Mm-hmm.
Yeah, the color pattern is being manipulated. And so what separates the control from the experimental groups in this case? What is the control in this case?
The brown.
The brown, and all of the experimental groups are
Colored or
all the
patterned.
other different color patterns. So if you were trying to describe to a new first year student who's trying to understand the scientific method and trying to understand what constitutes an experiment, how would you describe to them the difference between an experimental group and a control group? How do those two things compare with one another?
Control group isn't changing. Experimental is within the study.
So what is changed about the experimental group, and what is the same relative to the control group?
Just the variability.
I'm not sure what you mean by that.
I guess that would only apply to this specific study, is that the experimental groups are variable in
Yeah,
color.
that really only applies to
Yeah.
this. Let's think about something simpler. Let's take an example from class today. You have two different groups of people. One group of people you put on a diet, the other group of people you don't. You then look to see if the people on the diet lost weight versus the people not on the diet, not losing weight. What's the difference between those two groups?
The change in diet.
The change in diet. What's the sameness about those two groups?
Like the group of people. They're not the same group of people, but they're like pulled from.
They're not the same group of people, but you pulled them all from
Like a same pool.
the same pool of people, so that you don't put overweight people in the diet group and thin people in the non-diet group. You have a random selection of people in those two groups. So those two groups are the same as one another in all ways except the diet in this case. In this case, are the plasticine models all the same?
Except for the color pattern.
Except for the color pattern. So him talking about this in this way, he's going back to kind of the basics of scientific method and experimental design, where your experimental group is the same as the control group in all ways except for the variable that you're interested in, which in this case is color pattern. They're all, a centimeter in diameter. They all have an S-shaped pattern. They're all 16 centimeters long. The only thing, they're all made out of clay, and they're all fastened to the ground, and they're all non-movable, non-behaving models. They are the same in all of those ways except for the color pattern. And so if behavior mattered, which it might, it doesn't matter in the context of understanding these results, because the brown is just like the tricolor. The only difference is the tricolor is tricolor and the brown is brown. Mm-hmm. Neither of them have behavior. So once again, this statement, I've never actually talked about this particular paragraph in this way because nobody's ever covered it quite this way, but that goes back to the fundamentals of kind of how science works. And this just is an example that I had never thought about in that context.
Okay, so moving on to their potential, he touches on potential future studies. Of course, we inherently know what his next study was with Brody and Jansen. But there's one sentence here that he says the data here supports specifically this. And it's at the end of the first paragraph on page 234.
So you're saying that the brightly banded neotropical snakes are mimics of elapid coral snakes.
Mm-hmm.
Where are you at?
The last sentence, I think it's like the last sentence
Mm-hmm,
of the
last
first paragraph under Mimicry of Venomous Reptiles.
Oh, gotcha. Yep.
Yeah.
And the end of that sentence actually I think is more interesting.
Yeah.
Is that where you were headed, Mason?
Yes.
Yeah, continue, Elizabeth, to the end of the sentence.
Says that it strengthens Pugh's contention that this variety of banded patterns is evidence that deadly models protect a wide range of imprecise mimics.
So, expand on that. What is he exactly saying here?
So he's saying that like the mimics don't have to look exactly like the model. That's why it's like an imprecise mimic.
Mm-hmm.
But that they're still receiving the advantage and the protection from the deadly models.
Mm-hmm.
Because they're still being avoided.
Yeah. And we saw this in figure
5B.
5B, yep, exactly. So he's just reiterating the point here that the avian predators basically just see bright, colored, and banded and say, I don't want that. Yep. Right.
Remember we talked in class about kind of having simpler rules rather than complicated rules. Simpler rules are easier to deal with because they're simpler. You don't have to think a lot. And in the moment, as you're swooping down on a snake, as you get closer to it and you see more of the details of it, you have to either continue attacking, or swoop away. And if you have a simple decision tree for making that decision, that's much more effective than a more complicated decision tree. Wait, is that black and white with a red stripe, or is that just black and white? And is black and white? Oh yeah, black and white is the one that there's two snakes that are both black and white, and they're both venomous. And by the time you go through all that mental processing, you're dead.
Mm-hmm.
So yeah, the imprecision is useful.
Right.
Yeah, having a bunch of models makes imprecise mimicry work.
Yeah. So I guess moving into, I guess I could set the stage for Brody and Jansen, 95. The very last things he says, that this study establishes relative mimetic advantage varies among the protected banded patterns. So that's what we observed and already have talked about. Birds and predators see bright colors and avoid them. And so for further experimentation, especially at localities with only single model patterns, may elucidate this problem. So I was going to ask a question about future studies, but we kind of already know where the future went.
Yeah, sure. Well, so that's one thing, is to look at a mimetic system in which there are fewer models. What's a future study that I talk about in lab?
Was it the one in the Midwest?
Yeah, what goes on here in the Midwest?
There's a mimic, but we don't have any actual dangerous models.
Yeah, we have Lamprepeltis triangulum, one of the models, or one of the mimics in this study. It has a different color pattern, but we have a milk snake that occurs here in the Liberty area. I caught one down at Blue Springs Lake a couple of years ago. It's a white and black and red banded species, but that's probably close enough to be considered a coral snake mimic. And it occurs here in this environment where there are no coral snakes. So you have presumptive mimics in a place where there aren't any models. So the question might be, does that confer any advantage to the mimic in a place where there is no model? And one of the things that could be happening is that, and they talk about this a little bit in the Brody and Jensen paper, is it could be that since a lot of the predators of snakes are migratory in Missouri, it might be that birds fly from the north to the south, and hawks do this. There's an annual hawk migration to Central and South America. So the hawks fly to Central America in our winter, and then they come back in the spring, and they may take any experiences that they gain in Central America back with them, such that the milk snake here in Missouri may actually get a long-distance mimetic advantage. I'm not mimicking species that I'm sympatric with. I'm mimicking species that I'm allopatric with, but the bird that is trying to catch me and eat me is sympatric, at least for part of the year, with the model. And so I get that mimetic advantage remotely. Turns out by our data, that turns out not to be true. It would have been really kick-assly cool if it had been true, but it turns out that it's not true. Anyway, that is something that prior iterations of animal behavior students have discovered, and one senior thesis student. So, but that would be another avenue of future research. There's actually a situation on the East Coast that's been studied by a lab where at a much more local level, if you go towards the coast on the East Coast, you get into coral snake territory, and as you go up into the mountains, that coral snake isn't found in the mountainous regions, but you do get milk snakes. And over the course of maybe 100 miles or 200 miles, turns out that allopatric milk snakes evidently have a mimetic advantage, presumably that is gained by being in close proximity to coral snake range, but not actually sympatric with them. So there is something about distance. Once you get far enough away from the model, then the mimetic advantage goes away, whereas the mimetic advantage is present over shorter geographic distances. But that's another paper that we're not reading, because we're reading the paper that Avery is going to take us through now.
Okay, 1995 experimental studies of coral snake mimicry, generalized avoidance of ring snake patterns by free-ranging avian predators. I'm going to skip a bit of the introduction since we kind of already have some background information, but can you tell me where this study actually occurs?
It's a Costa Rican rainforest, right?
Mm-hmm.
And why is that?
Is it a rainforest?
Tropical dry forest.
It's a dry forest.
Mm-hmm.
So the
Is
first
that your
study,
question?
No.
Oh.
the first study was done on the Atlantic side, and you get warm, moist air coming from the Atlantic side, and that causes rainforests on the eastern side of Costa Rica. Then you go up over these mountains, you get a little bit of a rain shadow effect. Plus, the Pacific waters are cooler, and so that also leads to less rainfall. And so you have dry forests in western Costa Rica, and you have rainforests in eastern Costa Rica. So they basically went over the mountains to a drier environment. Sorry, how did we get on that?
She asked where it took place, and I
Oh,
said
gotcha.
rainforest. I was
Yeah.
looking at the wrong
Gotcha.
part of where I had it.
I think what you're getting at, though, Avery, is that pretty much the next sentence says that it's different from previous studies because this is inhabited by a single species of coral snakes and only a few mimics.
Yeah, so your study kind of focused on a 6-banded that were kind of sympatric to that region. In terms of this one, there's only one that's sympatric to that region.
Nice.
And then what are they trying to determine exactly in this study? And how would they do that? It's kind of at the bottom of that same paragraph.
Elizabeth, it plays off the question that you answered for Mason for his paper that was at the end of that paragraph on page 234, points at the reason that they're doing this one.
I think they're asking about, like, specifically about color patterns found in nature. I'm reading a sentence that says, none of these studies have demonstrated that free-ranging predators generalize their avoidance of coral snake ringed patterns to similar color patterns found in nature.
Yep. So they have their tricolor, which is kind of like native to that area, and then they're gonna introduce another pattern that's not. So kind of introducing like an experienced aspect on that portion.
So they have a control, one that's sympatric to predators around there, and then they have one that's basically allopatric.
Yep. And they're hoping to determine
Test avoidance of predators, yeah.
Because it's easy to see why there would be a generalized response on the east side of the mountains in Costa Rica, because why?
They're all there's
sympatric there.
There's
Yep.
many species.
There's lots of species, lots of color patterns, and so it pays in those environments to just say, it's bright colored, it got any kind of banding, I am not going for that. As opposed to on the west side, there's only one model, so you might expect that
That one
They
model
might
is
not
avoided.
know to be avoidant of others.
might not know to be avoidant of others, which also means that if you're going to be a mimic, you have to be
An exact mimic.
Precise.
a precise mimic versus an imprecise mimic
Mhm.
because there was only one model. And there's in the Brody and Brody paper, it talked about Poe's contention that a variety of banded patterns is evidence that deadly models protect a wide range of imprecise mimics. There's also this general view that as the number of models increases, the number of species that are mimics also increases. I don't remember if that was in the Brody paper or in the Brody and Jansen paper when
Say
they
that again?
talked about that. As the number of models in a community of snakes increases, the number of species that are mimics also increases.
I think in my paper it said basically the preciseness.
Then it may have been in this
They-
paper.
I wrote it in my lit some. It says relative mimic advantage decreases as similarity to the model decreases. Does that sound familiar?
Yeah, no, it's on page 187, this first full paragraph.
Oh, okay.
Investigations of coral snake mimicry in nature are hampered by two primary features of neotropical snake assemblages. First, most areas are inhabited by multiple species of ring snakes, usually including several species of venomous snakes with different color patterns. Maybe that wasn't the exact thing. Oh, well, at the bottom of the first page, a wealth of indirect evidence from parallel geographical variation in color pattern of neotropical snakes to the number of snake species that exhibit red coloration in sympatry with members of the genus Micrurus also suggests that there is some mimetic advantage to resembling venomous snakes. So there's this general trend. The more species of Micrurus you have, the more species of other snakes that have some form of red coloration in them there are. So that's this general trend. Sorry, now I feel like I've totally hijacked Avery. Take back over
okay.
from me.
In terms of methods, they're pretty similar. They make the same models with the soft plasticine so they can track attacks. So I won't really focus too much on that. And then
Ask
going-
Mason to revisit his treatment of what's a control and what's an experimental in this group.
What's the control and what's the
Mhm.
experimental in this group? Mason.
The control is the brown,
Yes.
and the experimental are the bicolor replica type and the tricolor.
Yes, and what did you say earlier?
I don't know. What did I say earlier?
You said the tricolor was the control and the allopatric was the experimental. The control and the experimentals are the same in this situation. The brown is the control. You then have maybe a secondary control, which is the tricolor, and then the experimental, because the experimental would be the allopatric
I thought I
pattern.
said the experimental was the colored models because they were changing, and the control was the brown because it didn't change. But then you
I thought you said
made sure
I
that.
thought you said the distinction was between the two colors, but I might have misheard. But anyway, yeah, let's just be clear about what.
Yeah.
It's difficult sometimes because sometimes studies have two controls. In this case, one control is the brown snake. That's a clear control.
Mm-hmm.
But the other control is kind of the tricolor because that's the sympatric.
It's
Because
maybe.
their question is asking, do these birds behave to allopatric patterns in the same way that they behave to the sympatric pattern?
Mm-hmm.
And that's a little trickier to think about from a strict experimental group versus control sort of group perspective. All right, keep going. You were talking about plasticine snakes.
Yeah, I just mentioned that the methods are pretty similar, except for the positivism. They're kind of going more towards is there an advantage to being sympatric or allopatric. Regarding figure 1, the first part tells us what exactly? The top graph A.
It just only shows the number of avian attacks on the control brown group.
Because
So, were there no attacks on the other two?
there weren't any attacks on the other two.
I actually had a question. Isn't that kind of weird though that there's nothing? Because I mean, even the other paper that there's expanded, there were, or would that be because there's a lot of different?
That's actually a
variations?
good question. Let's go back and look at are there differences in the number of replicas they put out?
Mine had 120 replicas per pattern.
Yeah, 60 in each transect. What was the situation with the Brody 1993 paper?
So, for experiment number 2, remember they had, where is that? Yeah, so 50 replicas of each pattern were placed on a natural back. Oh, no, that's the first one. Hold on. Where is it? I have it highlighted.
Yeah, 50. So it was very similar
Yeah,
in
50.
terms of sample size. Slightly more in the second paper. So yeah, that's a very good question. You know, is it weird that none of them were attacked? Considering that in the last study, you know, all of them were attacked to some degree.
But could you contribute that to the fact that there's only this one, like, dangerous species, and in that one there's a bunch of different mimics as well, so, like, at some point there might be some, like, reward to attacking, or some, not reward, but, like, a lack of consequences when you actually attack a mimic, and you
Yeah,
get, like,
so
So, like, here, like, you die no matter what, there's no advantage to attacking them. But
Yeah,
there
so-
could be a small percent.
So in the first paper, there were a lot of models, but there were also a lot of mimics. And so that makes imprecise mimicry effective, but it might also make aposematism slightly less effective. And so that might be a reasonable explanation. Because remember, the more numbers of mimics you have out there, the harder it is for aposematism to work, which is why here in the Midwest you see a lot of monarchs flying around, but you don't see very many viceroys. And it's because if there were a ton of viceroys, lots of birds would be eating orange and black butterflies, and most of the time they would have a good experience with that rather than a negative experience with that. Therefore, the act of being aposematic and distasteful in a monarch wouldn't really gain you much, because chances are you're going to have your wings ripped off anyway. It's only when the models get sufficiently abundant that having that aposematic color pattern works. But as soon as the number of mimics rises, then that dilutes the effect of that. And that might actually be what's happening, as opposed to on the west side. How many models do we have? One. How many mimics do we have?
Well,
Technically none, unless you
Is
bring
it
this
Yeah,
one.
I guess. That'd be
Actually, no. Well, they have mimics on this side. Who are the mimics? The potential mimics?
I'm guessing just mimics of the tricolor. They don't have any other coloring, do they? I don't know if they even mentioned it.
It does mention them. There's a whole portion of a paragraph that mentions them. It's right before materials and methods.
Oh,
Erythrolamprus.
And
By, what is it? By Bizona.
Lampropellicus.
Here's Erythrolamprus bizona. Lampropeltis triangulum you're already familiar with, hopefully. But I'll pull it up. Here's that one. We have Micrurus nigrocinctus. Micrurus nigrocinctus. And what's the last one? Scolophophus?
Scolophophus atrocinctus.
Scolophophus atrocinctus, okay.
And that one actually has a different color, doesn't it?
Yes.
Why didn't they use that then? Like as like another control or another.
It's the white and black banded with the red stripe down the back, which was in the previous study.
Is that a coral snake?
No, this is a
Is it dangerous at all?
nope.
Okay.
It specializes on eating centipede. So basically we have whoa. Here's a presumptive mimic. Here's a presumptive mimic. So there's that one. There's that one. They're very similar in color pattern. Here's a presumptive mimic. That is somewhat different, but it is white and black banded, which is a pattern that occurs on the other side of the mountains. And then here is the presumptive model. And it's a tricolor pattern, whereas the first two species were reverse tricolors. So what's the pattern that they are using here? They're using a tricolor pattern, and then what's the other pattern?
Bicolor.
Bicolor replicate.
Bicolor what colors?
Black and yellow.
Black and yellow.
Black and yellow. So they are incorporating the colors of one of the mimics, because it's black and white banded with a red stripe through it, so it's partly there. All right, so yeah, it is a little weird, but one of the explanations might be
Little weird what?
that there's nobody got attacked.
Oh, they're just avoiding.
Yeah, they're just avoiding anything
Mhm.
banded because in this environment,
It's
They're like hiding.
well, it's yeah. The allopatric one's foreign to the predators there, so they probably have never seen it
never seen it, so why avoid it?
because it could be dangerous.
Yeah,
or it looks similar to
it could be dangerous. It's banded.
yeah.
And what's one thing you know about snakes in this forest?
They're not more cryptic than others? Are you talking about that?
Well, so they didn't test crypsis, but they rely on crypsis
Yeah,
not
no,
being a
actually.
factor from before.
Yeah.
What do you know about at least one of the banded species in this forest?
Oh, it's dangerous.
It's dangerous.
It's dangerous, and it's dangerous enough. In the previous study, it was one of those with 2 pluses. Nigrocinctus is a reasonably big coral snake. You know, you want to avoid bandedness, and presumably that's what they're doing here. Do you have to worry about other color patterns in this forest?
Not
No.
necessarily, but it's not really something that they, like you said, think of. It's just kind of, they react to the bright colors and avoid them.
Yeah, so in this forest there isn't any variation. If it's banded, it's either going to be Nigrocinctus or some harmless thing. There's no harmful species that's banded with a stripe through it. There's no harmful species that is just bands at one end and the rest of the body is all one solid color, etc., etc. It's a simpler sort of stimulus. Like banded equals dangerous, and it might just be that simple of a decision tree in the mind of a bird.
Mm-hmm.
Continue.
In B, they mentioned that it's kind of non-avian attacks, but they also classify it as non-visual disturbances. Why do they make that distinction? Like, why is the non-visual part important?
Well, they mentioned that the number of alpacas were bitten by lizards and herbivorous iguanid. Am I saying that right?
I don't know.
Iguanid. It's a
Okay.
type of
Iguanid, okay.
iguana.
Kind of just gloss over the italics. They don't really.
Tinosaurus similis. It's a large.
well.
It's a large a large iguana with a crest down its back. Anyway, think iguana. Herbivorous. They're herbivorous. They're not going to be eating snakes. I don't think. Re-ask your question. I don't think it's been adequately.
Like, why is it important that like they separate those non-visual disturbances from assuming visual disturbances or avian attacks?
It's kind of like the same way they had it separated in the last one, right?
Yeah.
Of like they're just kind of ruling it out that these weren't attacks by birds, so they're not like relevant. They're
Yeah.
not. There's not a difference per se.
So there's no skewed data
Yeah.
for the actual avian
Yeah.
attacks.
And in terms of just being not predators as well, they're also not visual foragers, so they don't really have that distinction that these banded colors are dangerous.
What would be an
Mhm.
example of one of those? One of the group's organisms that made marks on these models?
I think they're talking about lizards.
Ants.
Yeah, ants.
Nidosauria.
Yep. That's exactly right.
Ants.
So they'll write a whole paper about Tinosaura, the iguana, that basically was feeding on their snake models when they are not predators of snakes. So, and they mentioned this in passing. So, visually oriented foraging in a natural population of herbivorous lizards in the Journal of Herpetology, and so they wrote a whole paper based on the anomalous data in this paper. What's going on? Oh, did it turn off? Hopefully it's still recording.
Do you want me to try to get to the end real quick?
Oh my gosh, we've
Yeah.
taken up a whole hour. Yeah, I guess let's try and get to the end. Well, actually, let's not.
Okay.
Let's just call it a day. We'll pick up here, make a note of where we are, and we'll pick up here at the beginning of class on Wednesday, and then at the beginning of class on Wednesday, once we're done with that, as soon as we're done with you finishing up, we'll move into the bat study. So I don't know if you saw. Well, you didn't see because the video cut off after 19 minutes for some reason. So I have to make a screencast of the rest of the time that we spent on Friday. But I want you to go and look up what is known about bats, how bats hunt for food, and how their prey avoid bats. There's a huge amount of work that's been done on this, and do that in preparation for Wednesday, because we're going to talk about a bat moth predator prey arms race. And that, in order for you to understand it, you need to know some basics about bat echolocation and about the anti-predator mechanisms that moths have to avoid bat predation.
So just how they hunt and how prey avoids them.
Yep. There's a lot of neurobiology involved, so we're going to be talking about the nervous system of moths and things like that. So, and you have a summary for me? Once again, what is it about a 2-page summary that you are failing to understand?
I went and, okay, so after the first, I got my first paper and I went back and made